Hello, everybody. Welcome to Creative Biolabs' presentation about oncolytic virus and virotherapy. Today, we are going to share some basic knowledge and the mechanisms of action about oncolytic virus. In the past decade, the strategy of cancer immunotherapy has gained prominence in preclinical development and clinical practice. Cancer immunotherapy now becomes to be the fifth pillar of cancer therapy, joining the ranks of surgery, radiation, chemotherapy, and targeted therapy. Traditional oncological treatments aim to directly remove or kill cancer cells, but receive high probability of recurrences or serious side effects. Targeted therapy requires identifying established oncogenic sites to limit the treatment scope. In contrast, immunotherapy uses certain parts of the body immune system to fight with cancer. It intends to enhance the host immune system's ability to eliminate cancer cells and contribute to tumor regression, anti-tumor immune memory formation, and ultimately endurable responses. Immunotherapy has great potential to cure cancer with very low side effect and high tumor elimination expectation. Immunotherapy includes adoptive cell therapy, immune checkpoints blockade, cytokine therapy, cancer vaccines, and oncolytic virotherapy. As one of the most encouraging therapy, now it's applied into treatment of multiple cancers. Here we take a closer look at oncolytic virotherapy. There has long been interest in developing an approach by which tumor cells can be selectively and specifically targeted and destroyed. Surprisingly, oncolytic viruses are viral strains that can infect and kill malignant cells without harming normal cells, while simultaneously stimulate the immune system and creating system anti-tumor immunity. Although viruses have been utilized as therapeutic agents in the form of vaccines since the late 1700s, their potential application as a cancer therapy was not explored until a series of earliest clinical references reports dating back to the early 1900s. The first case was in 1904, in which a 42-year-old woman with chronic myelogenous leukemia experienced a marked reduction in white blood cells during a flu-like illness. Later, a variety of different tumor types and viruses got involved in tentative oncolytic therapies, but interest in oncolytic viruses waned from the 1970s to 1980s due to the dreadful side effects. Until the 1990s, the development of genetic engineering in the 1990s made it possible to alter viral genomes. Viral therapy has achieved a leap from laboratory to clinical. Encouragingly, the adenovirus mutant H1N1 became the world's first oncolytic virus drug approved for cancer treatment in 2005 in China. A genetically engineered HSV became the first oncolytic virus approved by the FDA in October 2015 for the intralesional treatment of melanoma. Thus far, various oncolytic viruses have been successfully tested for their oncolytic activities against almost all types of cancers, and are being evaluated in numerous international phase 1, 2, and 3 clinical trials. The potential of OVs to improve the therapeutic ratio benefits from their ability to preferentially infect and replicate in tumor cells while avoiding destruction of normal cells surrounding the tumor, which is known as tumor tropism. There are two main mechanisms by which these viruses destroy tumor cells: direct lysis of tumor cells and indirect augmentation of host anti-tumor immunity. Tumor tropism is important for oncolytic viruses to function as a cancer immunotherapy. Both natural and engineered viruses take advantage of many differences of genetic characteristics and physiological features between tumor cells and normal cells, such as activation of proto-oncogene, inactivation of tumor suppressor genes, cellular defense mechanism defect, and abnormal growth signals. These differences bring about some different or specific expression of cell surface receptors, and exception of signal pathway. Owing to the rapid proliferation of tumor cells, a hypoxic environment formed is another worthy channel. Those characteristics can make the oncolytic viruses find tumor cells in the complex human system. Cancer cells have been shown to overexpress selected surface receptors. It is a core mechanism by which viruses may selectively bind to and infect cancer cells. Some viruses are able to pass through more than one receptor, and some receptors can promote the entry of more than one type of virus. For example, herpes virus completes cell entry through the herpes virus entry mediator, and selected nectins for cell entry. These surface receptors are overexpressed on some cancer cells, including melanoma and various carcinomas. Coxsackie can enter cells via CD54 and CD55, which can be overexpressed in cancers such as multiple myeloma, melanoma, and breast cancer. As a member of the enterovirus family, Echovirus has increased specificity for ovarian cancer cells by using the I domain of integrin alpha-2 beta-1 for cell entry, which can be overexpressed on these cells. Synthus virus targets cancer cells that overexpress the 67-beta-laminin receptor. This receptor can promote cancer cell invasion and motility. Measles virus utilizes the surface receptor CD46 for cell entry. CD46 normally functions to prevent cell elimination by inactivating the complement pathway of the immune system, and CD46 is often overexpressed by tumor cells. Poliovirus is a receptor that can potentially impair anti-tumor NK cell responses and is overexpressed by some tumor cells. It has enhanced specificity for various cancers through its targeting of CD155. Some viruses, like Newcastle disease virus and vaccinia virus, use endocytosis through membrane fusion and syncytia formation to enter cells. Certain oncolytic viruses, such as Seneca Valley virus and reovirus, are known to preferentially target cancer cells, but the cell surface receptor for entry has not been identified. Many of the oncolytic viruses in the clinic trial have a natural tropism for cell surface proteins that are aberrantly expressed by cancer cells. Oncolytic viruses can also target oncogenic pathways, which are different from the expression in normal tissues, thus maintaining sustained cancer growth. In cancer cells, cell cycle regulation and cellular proliferation are typically disrupted due to the activity of oncogenes and the loss of tumor suppressor genes. These changes can support viral replication and promote oncolytic virus-induced cell death. For example, the p53 tumor suppressor pathway is inactivated in nearly all human tumors. Some viruses, such as adenovirus, reovirus and parvovirus, preferentially target p53 mutant cancer cells because healthy cells with intact p53 undergo abortive apoptosis upon infection. Onyx 015 is an adenovirus mutant that lacks the E1b55K gene. It induces the degradation of p53 and, therefore, fails to degrade p53 during viral replication. On this basis, Onyx 015 is restricted for replication in normal cells, but is capable of replicating in p53 deficient tumor cells. PKR is known as the cellular immune system, which can prevent the replication of foreign virus infection. Both downregulated IFNs and activated RAS in tumor cells can block the activation of PKR, and make the virus successfully replicate in tumor cells. Oncolytic NDV and reovirus depend on this characteristic for their tumor cell selectivity. The retinoblastoma tumor suppressor protein pathway or PRBA pathway is dysregulated in a majority of human cancers, and it is targeted by the conserved region 2 of the E1A protein. As an example, the mutant ADZ1A gene was modified targeted the PRBA pathway. Another important way is the deletion of anti-apoptotic protein of engineered viruses, such as BCLXL. The viral genes encoding anti-apoptosis functions are essential for viral replication in normal cells. Deletion of multiple anti-apoptotic genes has generated tumor-selective oncolytic viruses including AD, HA-SVAO and VB. Aberrant activation of the WNT pathway is also implicated in driving the formation of various human cancers, particularly those of the digestive tract. CR adenovirus was developed to target this WNT pathway. When oncolytic virus successfully enters into tumor cells, how does it work? Oncolytic viruses are viral strains that continuously replicate in tumor cells and make the cells lysing, thus killing malignant cells. Recently, the focus in the therapy field has shifted from their direct oncolytic effect to their immune stimulatory effect. On one hand, after the tumor cells are infected, the oncolytic virus repeats in tumor cells and causes oncolysis. The release of viral progeny propagates the infection with the adjacent uninfected oncolytic virus and depletion of immunosuppressive cell types such as cancer-associated fibroblasts. Subsequently, dissolved cells release viral progeny, tumor-associated antigens including neoantigens, pathogen-associated molecular patterns and danger-associated molecular pattern signals, that is PAMPs and DAMPs. The tumor-associated antigens and neoantigens are released and taken up by antigen-presenting cells such as dendritic cells, which get involved in antigen processing and presentation, including increasing major histocompatibility complex class I and MHC class II expression on APCs. This process helps increase the MHC peptide TCR complex. The PAMPs and DAMPs can contribute to the maturation and function of dendritic cells, and stimulate the immune system by triggering activating receptors, such as Toll-like receptors which express on surface of NK cells and dendritic cells. In the context of the resulting immune stimulatory environment, collectively, these events result in the generation of immune responses against virally infected tumor cells, as well as de novo immune responses against DAAs and neoantigens displayed on uninfected tumor cells. On the other hand, upon infection with an oncolytic virus, tumor cells immediately initiate an antiviral response that consists of endoplasmic and genotoxic stress. This response leads to the upregulation of reactive oxygen species, that is ROS, and the initiation of antiviral cytokine production. ROS and cytokines, specifically type I interferons, are released from the infected cancer cell and stimulate immune cells, including antigen-presenting cells, CD8-positive T cells, and natural killer cells, thus causing a harsh environment for tumor cells. Some viruses demonstrate inherently oncotropic to target tumor cells selectively and more efficiently than normal cells, and trigger the anti-tumor immune system, resulting in tumor degeneration and health improvement. Other viruses must be molecularly engineered to selectively infect tumor cells. Advances in molecular biology have afforded the oncolytic virus field an opportunity to alter the DNA sequences of viruses and thus engineer viruses that are more specific for cancer cells than their normal counterparts. In general, viruses are chosen or modified to selectively target tumor cells, decrease pathogenicity to normal cells, decrease the antiviral immune response to prevent viral clearance, and increase the anti-tumor immune response. Although the mechanisms of action of oncolytic viruses are still incompletely understood, it appears that the therapeutic efficacy of oncolytic viruses is determined by a combination of direct tumor cell lysis and indirect activation of anti-tumor immune responses. However, further investigation of the underlying mechanisms is the driving force behind oncolytic therapy. Oncolytic therapy has made great strides in recent years. By embracing immune manipulation as a route to enhanced viral efficacy, oncolytic virotherapy is bound to rapidly expand in the near future. Creative Biolabs is a world-renowned service provider for immunotherapy. Taking advantage of the OncoVirapy Platform, Creative Biolabs provides customized, standardized, and reliable and high-quality oncolytic virus therapy development services for clients globally. Our service covers all aspects from virus engineering, cell biology to animal testing, with a special emphasis on virus engineering and in vitro and in vivo validation. Various types of oncolytic viruses engineering systems have been established in-house to facilitate oncolytic virus development with a fast turnaround time. GMP-compliant manufacturing of oncolytic virus is also included in our services. Thank you for watching our video. If you have any questions or need more detailed information about oncolytic virotherapy, please do not hesitate to contact us by email or visiting our website. We are more than happy to help you with your oncolytic virus project.